Hello to all the intellectual people who want to learn about the development of the I in evolution. So today we're going to learn about how evolution is constrained by the laws of physics, and that led to the development of the I in many different species. So eyes did not appear suddenly, they emerged through incremental physical steps, like I said, constrained by physics. And it started with photoreceptive molecules, which are composed of something called rhodopsin, which rhodopsin is made up by a protein called opsin, and retinol, which is a small chemical that fits inside the opsin protein. And whenever it absorbs light, it changes conformation, which can be read by the opsin protein and plays a role in the signal transduction of sight and vision in the rods and cone cells and in the eye, which eventually gets transduced to the optic nerve and the occipital lobe so that we can experience sight. But sight has evolved in many different forms of life. So you have eye spots, which evolved in basic organisms like the planarian, and then you have cup-shaped eyes and pinhole eyes, like it in the nautilus right here, and then you have lens eyes, which is like the ones we have. So this repeated emergence is a classic case of convergent evolution because in many different ways, mutational evolution has been guided by physics to create all these different eyes because at the end of the day, it's trying to make a system that can visualize what the organism needs to survive. So it's using light to do that. So you would assume that you'd have convergent evolution because it's all evolving to try to take advantage of that physics property, which is a field of photons. So once biology discovered light-sensitive molecules, the physics of optics and self-organization of biomolecular matter made eye-like structures almost inevitable, which I previously described. So to talk about the constraints of physics a little bit more, once organisms evolved photoreceptors like opsin, geometrical optics began constraining what works. Important physical constraints include resolution versus size, sensitivity versus resolution, the field of view, and optical material. So dense protein phases like liquid-liquid phase separation or crystalline assemblies satisfied these constraints. And I just have a diagram right here showing how um, evolution under constraints is kind of guided, even though it is randomly acquired traits through mutations that can still be guided by the laws of physics, which kind of allows only certain structures and morphologies to evolve. So let's talk about liquid-liquid phase separation. I talked about this a lot in my previous videos, but just to go over it again before I tell you exactly how it plays a role in eye development. So you can have proteins dispersed in a liquid, and then those proteins can form droplet-like phases. So it's going to organize water in a more hydrophobic lattice so that everything forms together, almost similar to like when water forms ice, it forms a lattice, but this is just a liquid form of organization where the proteins make the water form a certain phase that separates it from all the water around it. So this is called liquid-liquid phase separation, where you have two different phases of liquid, a more dispersed and disorganized phase, and then just a slightly more organized phase of water, which is created creating this lattice structure with the proteins, which forms structures and separates it from the dilute phase of proteins. So this is from a paper I published in 2022, and it just shows how these liquid phases these liquid phase separated organized condensates can actually guide fibers like collagen and fibronectin in breast cancer cells. So this is not only how breast cancer metastasis works, but also it plays a role in embryogenesis when cells need to move around and and guide fibers, and it also plays a role during embryogenesis in eyesight. Give me a second. So you can imagine that during the development of a fetus, you need all the fibers like collagen and fibronectin to be moving around and organized, especially during the development of the eye, where you need these small microscopic structures to finely organize so that they can detect light. So we'll get into more how that works in future slides, but the basic way that it works is that these receptors and these fibers are organized by these liquid phase separated protein lattices, because those protein lattices can bind to these proteins like collagen and fibronectin and beta 1 integrins and other integrins and other receptors, because they can bind to sugars. So those receptors have glycoconjugates, so that's branched in glycans, so they have branched sugars coming off these proteins. And the collagen and fibronectin can also have these branched sugars, which can bind to the proteins that are forming these liquid phase separated condensates and get organized by them. So everything is organized by the liquid phase separation being induced by the proteins and the glycoconjugates, which are the sugars, the branch sugars attached to the proteins. And you can imagine that the more proteins you have and the more branch sugars that are present, that it would promote the formation of these liquid phase separated condensates, which could then organize receptors and fibers close to the membrane in certain patterns, which plays a role in cancer metastasis. It plays a role in just the normal maintenance of tissues by fibroblasts. It plays a role in embryology. So it plays a role in many different aspects of biology. So now let's talk about liquid phase separation specifically in eye development. So I got this image from a paper called Phase Separation Pathways of Chiral Macromolecules at Liquid Interfaces. So this is just showing that chiral molecules, which is the mirror image of two molecules, like a right hand and a left hand image, even though they are very similar, they can still have different properties when it comes to liquid phase separation. So you can look at the liquid phase separation of two molecules, PLLA and MPLA. And when you have more MPLA, you'll have these eye-like morphologies that form. And when you have more PLLA, you'll have this crystallization dominant morphology that forms. But both of these types of liquid phase separation play a role in eye development. So liquid-liquid phase separation provides a soft matter mechanism for generating organized optical materials inside cells. In eye tissues, this matters because many structures, like lens proteins, must be extremely dense yet transparent. Phase-separated proteins can maintain high concentration and short-range order. So that's going to play a role in the refractive power and avoid scattering, so it can guide that light where it needs to be to play a role in signal transduction, so that we can eventually use that light to form some images in our brain and we can experience them. So the vertebrate lens is packed with crystallins, which is shown right here. This is crystallins. And the fact that they form this oligomer in a pentamer will help them form lattice structures, which can organize through liquid-liquid phase separation. In the previous image from my figure showing cancer cells, that was of the galectin-3 protein, which can also form pentamers through oligomerization, and can also organize in a lattice to liquid-liquid phase separate and organize these glycoconjugates on the membrane, like integrins that form adhesions on the cell surface, and also collagen and fibronectin, which which form the matrix outside the cell and form the dense fibers like collagen that connect your muscles to the skeleton, and things and other fibers like that, like fibronectin, which are smaller than collagen and form the network around microscopic cells. So the vertebrate lens is packed with crystallins here, which behave almost like protein glass or dense liquid, maintaining transparency while achieving a high reflective index. And again, they can achieve a high reflective index because they organize water in a lattice formation during that liquid-liquid phase separation process, which organizes all these proteins and the water together in the lattice structure, which can sequester together more proteins and and be less dense in water, which would create the crystallization dominant form. So by organizing water either in dense or less dense environments in the liquid-liquid phase separated droplet, it can either be more gel-like or more crystal-like, which is going to have a different refractive index. So how does these liquid-liquid phase separated condensates form the eye shape in the fibular network? So I took this paper from Nature, and it's called liquid-liquid phase separation within fibril networks. And when I said Nature, I meant the journal Nature, not that I just took it from Nature. Um, so this paper showed how liquid-liquid phase separation can organize these fibrils into circular domains, which shows how these liquid-liquid phase separation condensates can organize eye-like shapes, whether it be eye spots or more complex eyes like the ones we have. So you can see right here how during the development of the eye, these liquid-liquid phase separated condensates can form in between collagen or fibronectin, which I talked about earlier, and then it could move around the fibers like collagen and fibronectin until eventually you have a circular formation and all those condensates coalesce together. So during development, cells must assemble highly ordered optical tissue. Lens fibers contain crystallins in extreme levels, concentration right here. Phase separation like interactions help maintain uniform refractive index gradients. Similar self-assembly occurs in corneal collagen where spacing between fibrils prevent light scattering. So that spacing in collagen is created by these liquid-liquid phase separated condensates in a mechanism similar to this, because that's working on a microscopic level where you're having very fine fibers organizing into a circular domain. So from a soft matter perspective, the eye is essentially an optical device built from self-organizing biomolecular condensates and ordered polymers. So in red would be the biomolecular condensates, and green right here would be the ordered polymers, like collagen and fibronectin. So how is all this guided together? How are these liquid-liquid phase separated condensates guided to organize the collagen? Well, a scientist at Tufts University, his name is Levin, Dr. Michael Levin, and he does a lot of research on bioelectricity. And he has a paper called Darwin's Agential Materials, the Evolutionary Implications of Multiscale Competency in Developmental Biology. This paper taught me a lot about biology and where biology is headed to in the future. So I highly suggest reading it. And what in one figure and in one of his papers, he showed that you can actually scramble, they call it a Picasso-like tadpole, so a frog embryo while it's developing, and you can scramble the face and it will still end up producing a normal frog where the eyes are supposed to be. So it'll form a normal frog just like a normal tadpole will form a normal frog. So somehow, even if you scramble up some of the face, somehow the eye still learns where to go, even if you scramble it during embryogenesis. So, how I think that's happening and how science is coming to understand these things is because everything is set through bioelectricity. And he's shown this in planarians, and you can look at those papers where he can change the ion channel distribution in planarians, which changes the bioelectricity in the planarians, which will change their morphology. It'll make them have two heads and instead of one. So that's basically showing in planarians and in frogs that bioelectricity guides development. And the way that I think it's working is because that bioelectricity can change the liquid-liquid phase separation condensates. So ions can guide how these phase separated condensates form, and then those condensates will change how the fibers move around and organize, which ultimately changes how the cells organize because the fibers are the matrix for the cells. And then that's basically how you have morphogenesis and the development of not only the eye, but many different organisms, from the microscopic scale to the cellular scale to the macroscopic scale like eyes, and the whole system and organism scale. So you have the bioelectricity, which is created by the ion channels, which are set by genetics. Those ion channels create different electrical potentials throughout the organism, which creates set points for those liquid-liquid phase separated condensates to organize around. For instance, you have electrical set points for the eye, whether you disorganize it or not. That's going to cause the liquid-liquid phase separated condensates to organize around those bioelectric set points, and those liquid-liquid phase separated condensates will guide fibronectin and collagen along with receptors on the membrane to organize together. which will ultimately organize tissue morphogenesis and create the different morphologies you see throughout organisms. So the eye can be viewed as an emergent optical organ whose development relies on phase-separated assemblies forming transparent reflective materials, while evolutionary diversification of eye types reflects the geometrical constraints of optics that repeatedly shape photoreceptive tissues into functional imaging systems across environments. So the different environments that these organisms live in are going to provide the constraints from the physics that are in these environments that allow or guide these different eye types and all the different types of life we see on Earth. So evolution is fueled by mutational variation. I'm not trying to say that mutations and randomness doesn't play a role. It does, but it's guided by the constraints of physics. So I hope you enjoyed this one and have a good day.